Good morning to everyone. I am Dr. INBATHAMIZH. I am Assistant Professor, Department of Biotechnology, Satyabama Institute of Science and Technology, deemed to be University. I welcome you all to this session of lecture on basic concepts of enzymology. I hope you know all our mechanisms and our body system itself is governed by various reactions which we call as metabolic reactions. As well, the reactions are involved and utilized in various other external processes also. Now, there are certain biomolecules called enzymes which are called as biocatalysts, and these are involved in catalyzing biological reactions. As we go back regarding the discovery of enzymes, the name has derived from a terminology called zymase. The zymase in turn refers to yeast because in yeast, that is the terminology because the enzyme itself was discovered from an alcoholic fermentation reaction wherein alcohol and carbon dioxide were synthesized. So long back when the enzyme terminology was coined, it was only based upon the microbial enzymes. Now, knowing about the basics of enzymes, the molecule which reacts with enzyme is called as a substrate. Now, the enzyme has got a very small portion on which and through which the substrate can bind. The enzyme, if you consider, a very small region called active site is there to which the substrate molecule can bind. So, this active site plays a major role in enzyme activity. Enzymes are basically proteinaceous in nature. They are colloidal by nature. When we say protein, they are very, very specific in their action. Active site of the enzyme is also highly specific. It is a three-dimensional entity where the amino acids are arranged in a particular sequence. And at this active site only various interactions can take place. Also, the water molecule can also be eliminated during the process of reaction. Active site is the main point or the region of the enzyme where the substrate binds, and this was formulated by two main models. One was called as the Koshland's induced fit model, the other one was the Fisher's lock and key model. Lock and key model, the name itself suggests the substrate can bind to the enzyme just like a key fitting into the lock. That's why the name is lock and key model. And when the key fits into the lock, definitely the active site has to be very, very rigid. That's why you cannot change the conformation when the substrate binds to the active site according to lock and key model. The other model is the Koshland's induced fit model. Here, the substrate can change its conformation as it can get into the active site of the enzyme. So, the binding of the substrate into the active site and the conformational changes in the substrate and the enzyme can take place simultaneously. Actually, the binding of the substrate is going to induce some conformational changes in the active site so that the substrate can bind into the active site. So, this model is called as an induced fit model, and since the substrate can bring about the changes to the active site of the enzyme, this is a flexible type of model. Now, coming back to the properties of enzyme are highly specific in nature. When we talk about the specificity of the enzyme, there are a number of specificities we can signify based upon how the enzyme is going to act. The basic thing is the substrate specificity, where the enzyme needs a specific substrate to interact. For example, urease. Urease enzyme can act only on urea. So, the substrate is highly specific for the action of the enzyme. It can be group specific also. For example, lactate dehydrogenase can catalyze the reaction between lactate and pyruvate, wherein lactate can also be a substrate at times and pyruvate can also be a substrate at times. So it is a group specificity. Then it can also be optically specific, wherein the particular optical isomer alone will be catalyzed by the enzyme. For example, arginase. If you take arginase, it can act only on L-arginine, not on D-arginine. So it is highly specific on the optical isomer. Similarly, geometrical specificity, wherein the substrates, whether they are cis or trans, should be highly specific for the enzyme action. For example, fumarase activity. Now, fumarase can act only on fumaric acid, which is a trans isomer. It cannot act on a malic acid, which is a cis isomer, and the product is malic acid. So it depends upon the configuration and the isomers. So all these examples further justify that enzymes are highly specific in nature. Now, we'll move on to the nomenclature and the classification of enzymes. Enzymes, now they can be named based on the substrate, because substrate play a major role in the enzyme action. So based upon the substrate, the enzymes can be named. As earlier I've mentioned, urease is acting only on urea. Similarly, amylase can act on amylose. So the name of the enzyme is derived from the substrate. So the naming can be based upon the substrate. Next, it can be based upon the action, the reaction it can bring about. For example, if I say transferases, these enzymes can act and bring about transfer of one group from one molecule to another. So these enzymes are involved in transfer reactions. So the enzymes can be named based upon the type of reactions they can bring about. Some cases, the enzymes are also named upon not only the substrate involved, but also upon the reaction they bring about. For example, glutamic dehydrogenase. When I say glutamic dehydrogenase, the glutamate as a substrate, but also the reaction it is going to bring about, which is dehydrogenase. So the enzyme is named as glutamic dehydrogenase. Not only this, the chemical groups present in the enzyme, for example, phosphate, sulfur group, based on all these groups also, the enzymes can be named. For example, phosphoenzymes, phosphatases, sulfatases, all these indicate the group that is present on which the enzymes can act. As well, the enzymes are also named upon their chemical composition. For example, sulfur-containing enzymes, iron-containing enzymes, flavoenzymes. So all these, they indicate the particular chemical composition of the enzyme. So not only these, but the overall chemical reaction the enzyme can bring about. Based upon this, we have different categories of enzymes. For example, oxidoreductases. Now these enzymes, they catalyze redox reaction, both oxidation as well as reduction. Then we have transferases. As earlier I mentioned, they can bring about transfer of one group to another, especially transaminases, wherein in an amino acid, the amino group is transferred between two molecules. Then we have hydrolases. Hydrolases, they bring about the catalysis of a reaction wherein water is involved to break up the molecule. That is why it is called as hydrolase, which involves the role of water to split the molecule. Then we have ligases. Ligases, they bring in two molecules together, or we can say they join two molecules together. They are called as ligases. Another group is called lyases. Lyases is similar to hydrolases, but here it differs in not involving water. It can be the splitting of the molecule without the involvement of water. So altogether we have oxidoreductases, we have transaminases, we have hydrolases, we have ligases, and then lyases. So all these are the different types of enzymes based upon their overall chemical reaction which they bring about. Thank you for the patient listening, and we'll meet back again with another interesting topic. Thank you.